Hello everyone, Mr. Linder here. Let's talk about how the immune system combats bacteria and bacterial toxins. So the immune system is going to use non-specific and specific defense mechanisms to fight against bacteria and bacterial toxins. The non-specific defense mechanisms are designed to keep the bacteria from entering the extracellular fluid. The best examples of the non-specific defense mechanism are going to be things like skin and mucous membranes. Skin and mucous membranes form a protective barrier, and they're the first line of defense to keep bacteria from entering the extracellular fluid. Some other examples of non-specific defense mechanisms are going to be tears, saliva, acid production, and even the inflammatory reaction. When the protective barriers fail and bacteria enter the extracellular fluid, this usually triggers inflammation in the body. And inflammation is essentially a reaction that activates many cells to get rid of the potential pathogen that is entered into the body. Let's take a look at the inflammatory reaction. So the inflammatory reaction is really characterized by four cardinal signs: redness, heat, pain, and swelling. However, there's a fifth sign that happens, and sometimes that's the possibility of temporary immobilization of a joint. So what's going on in the inflammatory reaction? Well, when there's tissue injury, during the inflammatory reaction, there's going to be release of inflammatory chemicals. Some of the well-known inflammatory chemicals are histamine, complement proteins, kinins, prostaglandins, and others. There's also a release of leukocytosis-inducing factors. And basically, those are chemicals that are going to increase our white blood cell count. So during the inflammatory reaction, we're going to see white blood cells getting to the injured area. So what are the inflammatory chemicals doing? Well, the inflammatory chemicals are actually vasodilating arterioles and increasing capillary permeability, as well as attracting lymphocytes to the injured area. By vasodilating and increasing capillary permeability, you can increase blood flow to a damaged area, and that increased blood flow is going to lead to the symptoms of heat and redness, but that increased blood flow is bringing oxygen, nutrients, and more inflammatory chemicals to the injured location where there's potentially bacteria that have entered into the body. And so if we can bring the white blood cells and the plasma proteins that are going to fight against bacteria, we can stop bacteria before they enter into the extracellular fluid. We typically feel pain and we see swelling because the edema that's happening in the injured tissue space is going to be putting pressure on the neurons within that tissue space, and that's going to send signals to the brain that there is pain in that particular area. The white blood cells that have reached the injured location are going to be involved in phagocytosis, and they're also going to be involved in releasing more inflammatory chemicals, and also going to be involved in some of the specific immunity that takes place to combat against bacteria. The specific defense is typically seen by lymphocytes. Lymphocytes, or B cells, will become plasma cells, and plasma cells secrete antibodies. So the plasma cells that are attracted to the infected area can produce specific antibodies that can fight against a particular type of bacteria. So what we're looking at is the antibody structure. And so these B cells, these plasma cells that produce these antibody proteins, can be used to combat specific antigens. And so in this case, the specific antigen is the bacteria or potentially bacterial toxins. So what are some of the functions of antibodies? Well, antibodies can actually the antibodies can actually work to help activate more lymphocytes, and activating more lymphocytes is going to help produce more antibodies. The antibodies can also act as opsonins. Opsonins are proteins that can tag a particular bacteria and actually make it easier for phagocytosis. So other white blood cells can actually come and engulf the bacteria and prevent the bacteria from getting into the extracellular fluid. We can also use antibodies to block bacterial toxins. And so we can clump and inactivate toxins in cells and make it, again, easier for phagocytosis of other white blood cells in the inflamed tissue area. Antibodies can also activate antibody-dependent cellular activity. So antibodies can help activate natural killer cells or eosinophilic cells, and so can be used in other forms of immune responses. Antibodies can also activate the complement system. The complement system is a series of proteins that are produced by the liver, and they circulate in the cardiovascular system and are involved in immunity. And then antibodies also can affect mast cells and can trigger the release of chemicals from mast cells that also can help in the inflammatory reaction. So we can use non-specific and specific defense mechanisms to fight against bacterial invasion. A summary of fighting against bacteria is shown in this particular diagram. And so what I'd like to do is go through some of the events that are taking place to fight against bacteria in our immune response. So as you can see, there's an integration of responses that are taking place when we are trying to combat against bacteria or, again, bacterial toxins that have made their way from the external environment into the body. So here we have damage to tissue, and we can see that there's a bacterial invasion. So one of the things that we see taking place is the activation of these complement proteins. And again, complement proteins are liver proteins that circulate in the blood plasma and can get into the tissue spaces and can help us fight against bacteria. One of the things that complement proteins can do is they can trigger chemotaxis. And so complement proteins involved in chemotaxis are going to basically be signals that are attracting leukocytes to the injured location. So we see leukocytosis, the production of more white blood cells. We see the opening up of the blood vessels and the sticking of white blood cells to the blood vessel walls so that more blood cells can get into the injured tissue spaces. The terminology is margination. Margination means to stick to the capillary walls, and diapedesis is where the capillary walls open up and the white blood cells can make their way into the tissue spaces to aid in inflammation or to aid in specific immunity. Other fragments of the complement system act as opsonins. And so opsonins are like antibodies. Antibodies can coat bacteria, but complement proteins can coat bacteria. So these are proteins that can bind to a bacteria, coat the bacteria, and then make it easier to get rid of those bacterial cells, typically through phagocytosis. So by tagging the bacteria, it makes it easier for phagocytic white blood cells to come along and engulf them and get rid of the bacteria. Some other things that complement do is complement can trigger mast cells, so we can activate mast cells. And when mast cells are activated, and even basophils, other types of white blood cells, but when these cells are activated, they can release more of the chemotaxic agents, and they can release more of the vasodilators that we see in the inflammatory reaction. So in this diagram, it shows histamine as one of the inflammatory chemicals. And histamine is a vasodilator that increases capillary permeability, and it's going to help more leukocytes get to the injured area, and it's going to increase blood flow and bring oxygen and nutrients and plasma proteins. Here we have in this diagram plasma proteins. We can bring all of those things to one injured location, and so we can have this enhanced inflammatory reaction to fight against the bacterial invasion. The last thing that we see with complement is complement can activate something called the membrane attack complex. And the membrane attack complex is essentially the terminating part of the complement system. And what happens is these complement proteins insert themselves into the bacterial cell wall. And when they insert themselves into the bacterial cell wall, it basically pokes holes in the wall of the bacteria, and it leads to lysis of the cell. So it would look something like this. If we were looking at the inflammatory response and we're looking at phagocytic mobilization, we can see that the complement system and the inflammatory chemicals are bringing white blood cells to the injured location. Here we have margination. So first we have leukocytosis producing more white blood cells. Neutrophils as a good example. Margination is the sticking of the white blood cells to the capillary walls. And then we have diapedesis, which is squeezing the white blood cells into the injured tissue spaces. And how do they know where to go? It's because of chemotaxis. So those complement proteins and these inflammatory chemicals are signaling where these white blood cells need to go. The membrane attack complex looks something like this, where these complement proteins insert themselves into the bacterial cell wall, thus creating the holes in the bacterial cell wall, which is going to lead to lysis of the bacteria. And so that's a non-specific way to get rid of bacteria that have invaded into a tissue space and potentially could be pathogenic. This is a great summary of some of the stuff that we've been talking about, and I'm going to be talking about more of these chemicals. But if you want to use this as a summary, you can see that a lot of the vocabulary is on here as we talk about non-specific and specific defense. So to define what chemotaxins are, to define opsonins, antibodies, the complement system, histamine, and so forth. So a great summary of a lot of the vocabulary that we're going on in this discussion. So what else is going on to combat against bacterial invasion? So we've looked at so far that as the bacteria get into the injured tissue space, we've looked at the complement system and how that complement system can affect our white blood cells and can affect how, again, plasma proteins are getting into the injured tissue spaces. So what are some of the other plasma proteins that enter into the tissue spaces? Well, some are called acute phase proteins. Acute phase proteins are basically more proteins from the liver that will increase production of opsonins, and they will be things like C-reactive protein or anti-protease proteins that will assist in the opsonization process, the tagging of bacteria, to ensure that the bacteria are phagocytized properly. Some other chemicals that we also see are interleukins. Interleukins are actually released by white blood cells. So when you see white blood cells on this diagram, you can refer to interleukins, or IL for short. Interleukins are cytokines, chemical signals from cells that are secreted by leukocytes to primarily act on other leukocytes. They can work on other tissues, but they primarily work on other leukocytes. So they're communication signals. And there are many different types of interleukins. A good example of an interleukin is interleukin 1. And interleukin 1 is a chemical signal that can affect blood vessels. It can also activate the liver to make more acute phase proteins, and it actually can act as a pyrogen to trigger the body to run a fever, because that's a common response from bacterial invasion, is to run a fever to try to denature the bacteria. Another inflammatory chemical that is commonly seen to fight and help against bacterial invasion are the kinins. Kinins are derived from bradykinin, and these are plasma proteins that, when activated from bradykinin, they have vasodilating effects, just like histamine. And so they can increase capillary permeability, and they can help to bring more white blood cells and bring more plasma proteins to the injured tissue location so that we can fight against bacteria. Phagocytosis is a huge part of fighting against bacteria. And so bacteria that don't have some sort of protective capsule can be phagocytized immediately. And so phagocytosis is a big part of just engulfing bacteria. But if bacteria have capsules, capsules can actually hide bacteria from macrophages. And so that's why it's so important to have opsonin capabilities. And so those opsonizing capabilities come from complement proteins or acute phase proteins or even antibodies that are being produced by our plasma B cells. And so by opsonizing the bacteria, it makes it easier for phagocytosis. So again, examples of opsonins are C-reactive protein, which is a type of acute phase protein, antibodies, and the complement proteins that are going to help to get rid of bacteria that may be harder to capture by phagocytic white blood cells. We also have specific immunity that is going to work to fight against bacteria, and this is what we call the acquired immune response. In order to have an acquired immune response, you have to have some sort of activation of lymphocytes so that they can become specific for a particular bacteria. So if we already have antibodies against a particular bacteria, so if the antibodies already exist within our circulatory system, those antibodies can come into the injured tissue spaces and they can immediately start working against bacteria. So they can act as opsonizing agents, or they can act to neutralize bacterial toxins. If we don't already have antibodies against a particular bacteria, then we're going to have to activate the specific immune response, or the acquired immune response. And that's going to be with antigen-presenting cells. Antigen-presenting cells will essentially engulf the bacteria, so APCs. They're essentially macrophages. They're going to engulf the bacteria, and they're going to present fragments of the bacteria to cells called T helper cells. And then T helper cells can activate naive B cells, basically uneducated B cells, and those B lymphocytes can become educated. They can turn into plasma cells and start secreting these new antibodies. So we either have antibodies because of previous exposure to a bacteria, or we will go through a process of activating lymphocytes so we can produce new antibodies to fight against the bacteria. So antigen-presenting cells can activate T helper cells, which will activate naive B cells, and the newly educated B cells will become plasma cells and produce the antibodies necessary to fight against this new bacteria that is in the body. What's nice is if we already have exposure to a particular bacteria, and what this diagram doesn't show is that we would already have memory cells. And so if there are memory cells that exist against a particular bacteria, those memory cells will quickly become plasma cells so that we can make antibodies at a faster rate. It's the difference between what we call primary immune response and secondary immune response. Primary immune response is going to be slower because it takes time to become educated against a particular pathogen so that we can start ramping up antibody production. Whereas in a secondary immune response, it's going to be faster because we already have memory cells that are able to recognize this bacteria as something that's already been in the body, and so we can start making antibodies much faster. The last thing we see taking place when we're combating against bacteria is that we need some sort of repair to the damaged barrier that has allowed the bacteria to get into the body in the first place. So if injury has damaged blood vessels or has damaged tissue, then we need to go through the mechanism for repair. So to fix damaged blood vessels, you're going to see a platelet plug form, and you're also going to see coagulation taking place so that we can fix any damaged blood vessels. So if this is the area of injury and there's actually some damaged blood vessels, I know there's a vessel down here, but I'm going to go ahead and draw some up here. If there's damaged vessels, we're going to have coagulation and repair of those damaged vessels. And then to fix the damaged tissue, we're going to have mitosis, and we're going to have the replication of cells. And then we're also going to have to trigger the production of scar tissue. And so platelet-derived growth factor is actually going to be released from injured tissue to activate fibroblastic cells so that we can develop new connective tissue and produce scar tissue in the damaged area. So these are some non-specific and specific mechanisms for fighting against bacterial invasion or to fight against bacterial toxins. I hope that summary helps. Take care.